Have almost 161 participants right now. So
Rose. It was nice. Thank you very much for moderating this session. It was great. So we just have a short break of 5 to 5 minutes. We'll get back again because we have very exciting session. Next session is keynote.
We start with Dr. Shiv Pillai, who is a professor at Dr. Shiv Pillai actually wears many hats. So he is a professor of medicine and health sciences and technology at Harvard Medical School, Boston, USA. And he's also uh, the program director of an NIH-funded autoimmune center of excellence. And he has been the recipient of a number of teaching awards at Harvard, including the Irving M. London Award for Teaching, the Thomas McMahon Mentoring Award, and has been listed on Harvard Crimson's Professors of the Year. We have known him as somebody who made very fundamental contributions to immunology. He coined the term surrogate light chains for proteins that he identified as part of a pre-B receptor and that drives early B cell development. His laboratory postulated and provided evidence for the first ligand-independent signaling model during lymphocyte development and showed that BTK, the product of gene mutated in X-linked gamma globulinemia, is functionally linked to the pre-B receptor and the B cell receptor. BTK inhibitors now are finding usage in lymphoid malignancies and autoimmunity, and I think also somewhere in COVID management as well. Apart from the pro-B to pre-B cell transition, his group developed the concept of the follicular versus marginal zone B lymphoid cell fate decision. I remember several discussions at ICGAB in Dr. Kanuri Rao's group mentioning Shiv Pillai's work at that time on this subject. And he has recently identified a metabolic transitional pathway to follicular B cell transition that is blocked in a human uh, common variable immunodeficiency and identified a block in T follicular helper cell development in COVID-19 that prevents the formation of germinal centers. His laboratory has also linked cytotoxic CD4 T cells in endothelial cell apoptosis and systemic sclerosis and to mesenchymal cell apoptosis in IgG4 related disease as a major step in the development of autoimmune fibrosis in these diseases. And we all know he is the co-author of a monograph. He's the author of a monograph on lymphocyte development and co-author with Abul Abbas and Andrew Lichtman, very widely used textbooks of immunology. I must take this opportunity also to thank Sunil Arora, Amila Basti, FIMSA, and IAS for organizing this very wonderful lecture webinar or series. And also, I thank them for giving me this opportunity to moderate this session. Today, Dr. Pillai at this session will talk about, I think, approaches to human immunobiology, and I leave it to him to tell us more about this thing. Dr. Shiv Pillai, please.
So thank you, Pawan, and thank you, Sunil and Amit, for the invitation into FIMSA. So I'm going to share my screen, and hopefully you can all see it now. So is this visible to you? Yeah. Okay. So today I'm broadly going to talk about approaches to human immunology. I was also asked to just broadly cover some overview of immunology, but I've noticed that you've had a very good coverage today of innate immunity, and there was probably a wonderful discussion on the thymus earlier in the day. So I'll just touch briefly as an overview, and I'll come back to it maybe in some other form at the end of the lecture. So we all know that we have innate and adaptive immunity. Innate immunity has barriers, all the cells you've been hearing about, complement, type 1 interferons, pro-inflammatory cytokines. But adaptive immunity is mainly mediated by B cells making antibodies and T cells, which can either help other cells or can be cytotoxic. Okay, and innate immunity is immediate, adaptive immunity is late. Innate immunity, we break into inflammation and the antiviral state. you can neatly divide it into these two halves. And you've heard a lot about inflammation today. Inflammation comes in different forms in disease. This is just acute inflammation that's depicted here. This is what we use when we think about inflammation soon after an infection. But if you're thinking about a chronic disease, there's more to it. So when you're studying human disease, you actually want to know what is the nature of the inflammation, what's driving it, what is the adaptive component that's triggering it if there is an adaptive component. And so this is one half of innate immunity. The other half of innate immunity is the antiviral state. This is, of course, very important in COVID-19. Now we realize that, because the antiviral state depends on the generation of type 1 interferon. And tomorrow, when I talk about COVID-19 and some of our work, we'll briefly discuss how this is subverted by the virus. So type 1 interferons bathe other cells, signal through the type 1 interferon receptor, and induce in these recipient cells an antiviral state. And type 1 interferons also affect adaptive immunity in NK cells, and so on. Okay? Now, we think about the sentinel cells of innate immunity as being mast cells and macrophages, the early for inflammation. So mast cells and macrophages, and you heard all day about TLRs and the activation of these cells. And then we also have the dendritic cell, which is going to give us the link between inflammation and adaptive immunity. And the key thing to understand, of course, conceptually, is that in order to activate adaptive immunity, we need both something to be foreign, to be recognized, and then we also need to have a signal that tells the T cell, if it's starting with a T cell in the lymph node, that this dendritic cell has come from a land of danger. So if the dendritic cell is activated by a TLR, it will have the ability to provide a second signal. So normally, let's assume you had a foreign protein, got taken up by a DC. Maybe there's a peptide, this brown peptide presented on class 2. It could be recognized by the T cell receptor. This is foreign, and it can only provide signal 1. This is not enough to activate a T cell. We always need a second signal. So TLR activation will induce a co-stimulatory ligand like B7. B7 will trigger CD28 on the T cell. So this is the naive T cell in the lymph node. And now we will have activation of the naive T cell in the lymph node. Okay? The other thing we want to remember, of course, is that when a cell captures, a dendritic cell captures a pathogen antigen and then gets activated through TLRs, it migrates through afferent lymphatics to the lymph node, provides these two signals, one for foreignness, one for danger, activates T cells, and these T cells now become effectors. And it is the effector T cells which are going to go and migrate back to the site of inflammation and deal with the pathogen. And these effectors, some of them will become memory cells, the majority of them will die, and that's called contraction of the immune response. So we make immune cells in central lymphoid organs, we generate diversity of the repertoire in central lymphoid organs, we have central tolerance in central lymphoid organs. Naive cells only go to lymph nodes and to spleen and Peyer's patches, so secondary lymphoid organs. That is where naive cells get activated, and they need to get activated by two signals. Okay, and then these naive cells got activated, they migrated back, went to the site of infection as effectors, dealt with the pathogen, and then maybe some of them become memory cells. And then again, just broadly speaking, I'm actually going to talk about a CD4 subset in the context of disease, where I try to explain approaches to human immunology. I just want to tell you that the classical thinking, even in our textbooks, is that we have Th1s, Th2s, Th17s, TFH, Tregs, and if Amit is in the room, always Th9 as well. But there are some more subsets to think of. And certainly today, I'll talk a little bit about the one that might be linked strongly to disease, about which we're learning more. Okay, so if you think about disease, and you think about how we consider disease and how we divide disease in general, there's a few categories we can break everything into. Okay, so we have Mendelian disorders, single gene disorders, relatively easy to understand. Infectious diseases, even COVID-19, again, you have some combination, like if you now know the story about type 1 interferon receptor pathway mutations, there can be a combination of genetics and a microbe which can cause a disease, and this would be a severe infectious disease. In cancer, again, relatively easy to understand, not that we can solve everything about it, is that a single cell acquires somatic mutations, and those somatic mutations allow the cell to grow in an uncontrolled manner. And everything else, which is the real frontier today, both in biology and medicine, is a complex disease. And complex disease is something that everybody on this Zoom call has in some form or the other. If they don't already have it, they'll have it in the next 10 years or so. And this includes type 2 diabetes, atherosclerosis, hypertension, asthma, allergies, autoimmunity. All of them are complex diseases. And we hand wave and call them multi-gene disorders. We don't fully understand them. Now, there are some occasional diseases, and these are 2 occasional tumors, where we actually have no driver mutation, but we have epigenetic changes. And these somehow will fit into the same category as complex diseases, okay? Because there's a lot of epigenetic alterations driving disease. So what do we think is happening in a complex disease? And again, I have to say, we don't know all the answers yet. Obviously, this is a frontier. There's some genetic susceptibility. There's no question there's some amount of it. It cannot explain everything. There's usually a trigger. Now, if this is not an infectious disease, then there still may be some change in the microbiome, some metabolites generated, and these are being studied. And there's very interesting data coming out. And even in the disease we study, we collaborate closely with Ramik Xavier to look at the microbiome. So either antigenicity shared with the microbe or a novel metabolite coming from a microbe might cause an epigenetic alteration in an immune cell. Then some clone of immune cells, which is altered, might then break tolerance and lead to enhanced inflammation, and this might be the cause of a disease. Okay, this is the way we think about it. And clearly, while we study COVID-19, we're going to be following patients because now we have an idea of what the trigger was, and then we want to see what are the consequences. The other point that I think we need to understand, when we study disease, we do have to study disease not just in the blood. Unless you're studying, just a leukemia, which is highly bloodborne, you do need to go into a couple of other sites. So if you want to analyze a human disease, one thing we have to do is to look at lymph nodes. And lymph nodes, as you know, have both T cell zones and B cell zones. And we need to be looking at lymph nodes in the context of disease, because the immune response that causes a disease is likely to arise from a draining lymph node. You might have factors in the tissue, but then the draining lymph node is important. And the other thing you want to consider, of course, is that there's a lot of things that happen only in lymph nodes. For when I talk tomorrow, we cannot look at germinal centers in the blood. You have to look at a lymph node. And I would say one step further, in any disease which involves an organ, you need to look at the organ as well. So to be comprehensive in the immunological approach to human disease, we need to be looking at many things from genetics to microbiome and so on. But we need to interrogate the blood. We need to interrogate lymph nodes when we can. And we need to interrogate the tissue, the organ in which the lesion occurs, because then we are going to get a better picture. And what I'll do today is we study a few diseases in my lab. So we study systemic sclerosis, we study IgG4 related disease, we study fibrosing mediastinnitis. This disease is actually caused by Histoplasma capsulatum. And it gives us some clues because we know there's a microbial cause to fibrosis. These are all fibrotic diseases. We actually look at some diseases in which there are single gene mutations. So we have a huge cohort which we have built together at Mass General, about 270 patients now, of patients with single gene mutations in the immune system. But we are focusing on those that cause autoimmunity, fibrosis, and which block B cell development. And we have some interesting stories. I won't have time today to get into that. And I'll talk about COVID-19 tomorrow. So today, I'll largely be focusing on the first 2 as examples to explain strategies for how you approach a human disease. So we think about the workflow for studying human disease. When you can interrogate blood. So, one standard approach we like to use is 25 color flow panel for T cells, 25 color flow panel for B cells. This allows you to purify cells, do RNA-seq, ATAC-seq, cut and tag. Single cell RNA-seq can be done from the blood. You can look at repertoire. Some people prefer to do site-off as the first thing. We reserve it for certain reasons. You can do antigen discovery. You can look for what is the antigen that in a disease a B or a T cell sees. And a tool that we've started to use a lot more with Willie Haas at Mass General is mass spectrometry. We can get 8,000 proteins easily from a specific immune cell. And then, so mass spectrometry is again unbiased. It's like single cell RNA-seq, but looking at proteins. And at the end of the day, you want to look at proteins. We can interrogate draining lymph nodes, and we prefer to use multispectral imaging approaches. We can do cell distance mapping there, but you can also use Hyperion, you can use Codex. There are other alternatives that some people might prefer. I just think that morphology-wise, we do better with what we're using. We can interrogate the target organ. So if I have a disease in which the skin is affected or the lung is affected, or maybe the pancreas is affected, I can go and look at those tissues. Again, we prefer to use this multispectral immunofluorescence, and you'll see more of it tomorrow when we do lymph node studies. But I would like to do cell distance mapping. We want to know which cell is talking to which cell. What is it making? And what's the frequency of that interaction to quantitate it. Single cell RNA-seq doesn't tell us that because you broke everything apart, but we like to use this as well. But you can use, obviously we use single cell RNA-seq as well. It's a powerful tool. Again, you can use other tools. Slide-seq is another tool. It hasn't actually worked out as well as advertised. Then we need to look at underlying mechanisms. What's the microbiome like? We do antigen discovery as mentioned above. You can look at the chromatin state of cells that are driving a disease, see what epigenetic alterations drive those cells. Then you can test the validity of any model you build. For this, you have to do clinical trials as one approach. Sometimes that will give you a clue. If you can, you model it in mice or in humanized mice. Okay? These are and you can use CRISPR, of course, to do a lot of the modeling. So what do we think you need? And if your disease is caused by T cells, we think there are some Koch postulates we can use. Are there clonal expansions of effector memory cells? If there are clonal expansions, then we can look at them in detail, and you can look at, prove it's a clonal expansion by next generation sequencing. I'll show you an example of that. If you've identified a T cell clone in the blood and you think it's expanded, is it in the tissue? If it's not in the tissue, what does it mean, right? It has to be, if it's causing the disease, it should be in the tissue where the lesion of the disease is. So very important to go look at the tissue. And then if you think that clone is driving disease, or those clones are driving disease, you need to look if therapy, any therapy that improves the disease, that it caused the loss of those clones. So if you fulfill these postulates, you have a better idea of what's going on. So I just want to put it out there. We're looking at a few fibrotic diseases. A lot of people die from fibrosis. There's so many different causes of fibrotic diseases. I'm going to first give you the broad model so that you can follow along, and then I'll talk about a couple of diseases with examples of how we interrogate disease. So this is our broad model, and it applies to 2 or 3 diseases now, at least 3 that we've looked at. And we think CD4 CTLs and sometimes CD8 cells are driving the disease. They are clonally expanded, and I'll explain what CD4 CTLs are in a bit. We think activated B cells are playing a role. They're contributing. Maybe they're presenting antigen. Maybe they're making things. I'll show you they can make enzymes. You would never believe that B cells are doing these weird things, but they do. We find some of these weird B cells in COVID. We just published that recently. I'll talk about briefly tomorrow. We think you then have the induction of apoptosis. So we think in every severe fibrotic disease, even in pulmonary fibrosis, which can have genetic causes, you have apoptosis. And some target cell is dying by apoptosis. And then when it's remodeled, space is created, you remodel, you over-aggressively remodel, you get fibrosis. So you have macrophages, fibroblasts, myofibroblasts all involved. But we think the main trigger here is immune-mediated apoptosis, setting the stage for now to replace those dying cells with a fibrotic tissue, often a fibrotic mass. So systemic sclerosis or scleroderma is a very bad disease. It reduces the lifespan of women who have it by 20, 25 years sometimes. And it occurs predominantly in women of childbearing age. If you see a patient, it's a very severe disease with affecting almost every organ you think of. But we worry most about things like the lung, the kidneys, the gut, and the skin. Okay. Now, there are endothelial alterations in this disease. And there's always been a belief there's some connection to the endothelium, and I'll tell you what we think is going on. The other disease that I'm going to use in parallel because it's almost like a model system to go and study systemic sclerosis is IgG4 related disease. Again, here you have a mass. Patient often presents misdiagnosed as having a tumor. Is actually just a mass. In that mass, it's inflammatory cells that have collected. You see blood vessels are occluded. It's called obliterative phlebitis. And you see storiform fibrosis. It's just a description for the type of fibrosis. For diagnosis, typically you have to say very high proportion of the cells, the plasma cells in the tissue, are plasmablasts. But the majority of the cells are actually T cells. You have T cells and plasmablasts, activated B cells of various sorts that we have looked at, all in the tissue. And it can affect almost any organ. So I'm just going back to some work we did when we started to look at IgG4 related disease, looking at the blood. We gated on effectors and we found, okay, yes, in the disease, there's lots of effectors here. And of course, in controls, effectors are a very small subset. You can't see them. We then looked at these effectors and characterized them transcriptomically and found that they're kind of weird, that these effectors all had a signature of a CD8 T cell. This is a CD4 effector. It has a CD8-like signature. And these cells have been described in different contexts, but I think we established that you can have disease because of these cells. And these cells express TBET, CD97, SLAMF7. They've turned on a program of CD8 cells. And I'll tell you towards the end how we think this has happened. Okay? So there's CD8 programs turned on CD4 cells. These cells are called CD4 CTLs. They protect against dengue. That's well described. They protect against other viruses, including CMV. They're induced in HIV, provide some protection, obviously not enough. And they also have been shown in cancer. And so tumor immunologists are now very interested. So both Jim Allison and Jed Walchuk have papers on CD4 CTLs in cancer. But we've looked at it in inflammatory disease. The numbers of CD4 CTLs in active disease goes up dramatically compared, this is all blood, compared to controls. Okay? So we asked, are these clonally expanded? And the way you look at it is you take, let's say you took out 50,000 cells and you want to ask, is this clonally expanded? You take those cells and we have rearrangement of the beta chain and alpha chain of the T cell receptor in a T cell. So we basically do next-gen sequencing and we'll may sequence 5 million times to ask, is there a clonal expansion? Are all these cells derived from 50,000 different cells, all the sequences? Or they derived from 10 different cells or 50 cells? What is the answer? So when we did that, so you can see this, these are 4 patients with, this is the one part of a study we did where we chose people. So remember, about 40% of all humans, or 30 to 40% have allergy. So we chose 4 patients with IgG4 related disease with a history of allergy because we can't find Th2 cells in the blood otherwise. We wanted a control. So these are patients who have got IgG4 related disease and they have allergy. We can purify the Th2 cells. This is the GATA3 positive cells. Those are the SLAMF7 positive CD4 CTLs. And what we find is CD4 CTLs are big clones. Whereas this is the history of allergy in this patient, the little dots. Okay, so there are big clonal expansions of CD4 CTLs in this disease. In the tissues, we can find these cells in the tissues. You can quantitate them. This is in a bunch of patients. And you can see that the dominant CD4 cell here is this CD4 CTL, and Th2s are relatively rare. Okay? In systemic sclerosis, given that there's a highly fibrotic disease, we did studies in parallel, and this is again something that was published earlier this year. And here we showed that in systemic sclerosis, of course, there's lots of CD4 positive, granzyme A positive cells, and very few GATA3 positive cells. On the other hand, if you look at bullous pemphigoid, which is really a Th2 dependent disease, there's lots of GATA3 positive cells, and they are Th2s. So if you quantitate again, the red bars are CD4 CTLs, the green bars are Th2 cells. So let me go back. And you can see that by and large, there are a few patients in whom, there are a few out of these 35 that we're looking at, there are a few in whom you do have good Th2 cells. But for the vast majority, when we quantitate it, the majority of the cells, the dominant cells are CD4 CTLs. This is different in bullous pemphigoid, where the dominant cell is the Th2 cell. And this is the quantitation over here. So in systemic sclerosis, you tend to have more CD4-CTLs than in bullous pemphigoid. We took 2 skin diseases, so we could do these comparisons. If we look at all CD4 T cell subsets, so we are able to stain all the cells in the same experiments, and we quantitate CD4-CTL, Th1s, Th2s, Th17s using different transcription factors, TFH and so on, and Tregs. You can see CD4-CTLs tend to dominate. So just by using quantitation, and as I'll show you in a minute, by looking at actual cell-cell interactions, we can make some conclusions. Say, yeah, these cells are the dominant cells, and they're in the tissue. And this is not in every disease. In certain diseases, the ones we're looking at, this is dominant. Then the question was, do these cells induce apoptotic death? And then this interesting result came out. We noticed that endothelial cells are undergoing apoptosis quite a bit in this disease, in systemic sclerosis. In IgG4-related disease, we have apoptosis, but not so much of endothelial cells. So these CD31 positive cells are endothelial cells. And you can see cleaved caspase 3 inside these endothelial cells. And the blood vessel cells, some cells are undergoing apoptosis. This is, I'm not sure how well you can visualize this, but here's an example of a CD4-CTL, which is granzyme A positive. Okay, it is actually in the vicinity of a cell over here, which is essentially this endothelial cell. This is the endothelium. But that endothelial cell has acquired a little granzyme A. So you believe the granzyme A has been delivered by the CD4-CTL into this endothelial cell. And that endothelial cell is undergoing, it's got cleaved caspase 3, it's undergoing apoptosis. And we've done this a few times. So I think we're even able to capture, if this is the endothelial cell here, which is dying of apoptosis. And this is the endothelium. You can see the circular form here. And the CD4-CTL has apparently executed it by putting granzyme A into it, and it now has cleaved caspase 3. So again, by we can use tools like cell distance mapping software, ask which cell is kissing which cell, quantitate it, and so on. So we see these interactions, strong interactions between CD4-CTLs and endothelial cells, and frequently apoptosis of the endothelial cell. And then we can quantitate it and say, this is what's commonly happening. So then if I give you a model for systemic sclerosis, the model says that cytotoxic cells, mainly CD4-CTLs, may induce perforin and granzymes, target some antigen, obviously very important to know what the antigen is, in an endothelial cell, and the endothelial cells now undergo apoptosis. In this context, there is some role, we think, for activated B cells in the process. And there are now some papers, even one from India, showing that rituximab is improving a little bit the outcome in systemic sclerosis, not a huge difference, not like IgG4-related disease. So there may be a role for B cells in the process. And at the end of the day, you will now get fibrosis. Okay, and this is fibroblasts and macrophages play a role at the end stages. But I think the key event now is immune recognition of an endothelial cell by a CD4-CTL. Okay, HLA-DR is expressed at high level in these endothelial cells. So during inflammation, this is what happens. So this also explains, so I didn't show you that for systemic sclerosis. I'm now showing you this for IgG4-related disease, where in inflammation, HLA-DR gets expressed on many cell types. But in IgG4-related disease, endothelial cells are not dying, but Vimentin-expressing mesenchymal cells are dying. So whatever the driving antigen is in IgG4-related disease, is not the same as systemic sclerosis, though the mechanism may be similar. Okay? Again, we have quantitation over here, and we're comparing Sjogren's syndrome here with IgG4-related disease, and you have a fair amount of mesenchymal cell apoptosis in IgG4-related disease. So a broad view now, obviously this is all human. We are based on comparisons, there's some heterogeneity, but we think systemic sclerosis is a CD4-CTL driven fibrotic disease in which the antigens are endothelial. IgG4-related disease is also CD4-CTL driven disease in which the antigens are stromals in fibroblasts and myofibroblasts. In fibrosing mediastinitis, we have actually shown this paper has already been accepted, it's not out yet, that the CD4-CTLs that are maybe driving the fibrosis, we can't prove that, but these CD4-CTLs are specific for histoplasma capsulatum, that we've shown. Okay? So in some cases, it may be an autoimmune fibrosis. in some cases need pathogen-induced fibrosis, but CD4-CTLs may be playing a role in the disease. Okay? Now, I said a lot about CD4-CTLs. How do we study these cells more? How do we put it all together? So we also we have been using epigenetic approaches and single-cell RNA-seq and other tools, which I'll show you a little bit about, to understand novel disease-related human immune cells. And if you get enough cells, you can do a lot of studies, and some of these tools can be used with very few cells. So most of you have heard of single-cell RNA-seq. I won't belabor this. You can use single-cell RNA-seq. You put one cell per droplet or one cell per well, get transcriptome data, and that's your single-cell data. Okay, and a lot of computation, not very complicated, but we do computational work to resolve it. This is some of our data. This is published now using single-cell RNA-seq, repertoire, flow cytometry to look at subsets of CD4-CTLs. And we can divide this into subsets. And the subset that causes disease and which expands and clonally expands is the CD4-CTL effector subset. We call it effector because we think it's driving disease. But there is a number of, there are 3 different subsets we can see by single-cell RNA-seq. Again, here you'll see by single-cell RNA-seq, this is just using time of flight. There are 3 different subsets. We put it in this context. This is actually by flow, we are looking at the subsets because you can actually characterize them, and then we can look at them in much more detail. We've done RNA-seq on them, looked at what's different between subsets, characterized these differences, and clearly the ones that we call effectors are the ones that we think are driving disease. We've correlated with disease. Now, none of your studies are worthwhile. None of our studies are worthwhile unless you have a good clinical correlations, good characterization of patients, subclassification, and we do all that to try to understand it. This is to show clonal expansion, which ones are highly clonally expanded, and you can see that over here, that the frequency, these are the cells in which you have the frequency of clones which are going, what is the top 10 clones in each subject, okay? This is what we see. Okay, how about epigenetic marks? So the way to think about this is, of course, that if a gene is going to be expressed, it's in what you would call in heterochromatin or in a closed chromatin state when a gene is, when it's not expressed. When it is expressed, you're going to have movement, you're going to have opening up of, so the nucleosomes are going to move apart, and you're now going to expose the gene to be expressed. Okay, so this is just a simplistic way of thinking about it. But one way to think about gene expression in a specific immune cell, and this has come from many people's work in the past, is that if you look for genes which are driving the biology of that cell, they will be regulated by regulatory elements we call super enhancers. And a super enhancer is nothing but the fact that you have just defined by size, how many binding sites are there for many different transcription factors. So a typical enhancer will have a few sites. A super enhancer will have many sites. It'll be a larger piece of open regulatory regions of a gene when it's expressed. This is the transcription start site shown over here. So if you can identify which are the genes in which there are super enhancers, that already gives you a clue which are the genes that drive the fate of that cell and drive the biology of that cell. It's just a tool to use. And basically, this is a slide saying the similar thing, that, a typical enhancer, you form this structure over here. It's a TAD or a transcriptionally active domain. And in the case of super enhancer, you have a lot of binding sites, a lot of transcription factors, a lot of protein-protein interactions driving this. And this is why this gene is expressed so much better in that particular cell type. So we use this as a tool. I'm not going into the methodologies of these. We could discuss that, but we use things like ATAC-seq, we use DNA methylation in order to basically to identify super enhancers. We use ATAC-seq and we use histone modifications. Anyway, so we identify genes linked to super enhancers in CD4-CTLs and some TFH subsets we're interested in. This helps us identify pathways and enzymes. Then the whole goal is, if we know more about this, then we can generate small molecule inhibitors and maybe new drugs to suppress immunity. Now, I'm going to just show you a little bit of our data. So this is, if we're looking for super enhancers, and we're looking in both CD4-CTLs and TFH cells. And in TFH cells, BCL6 comes up on top, which you will understand makes a lot of sense, because BCL6 is the master driver of this cell. And in CD4-CTLs, we come up with multiple things. And so these are the things we're investigating. This is just looking at differential exposure. We're looking at naive cells, TFH cells, CD4-CTLs. Just by looking at this, you can tell there's differences in what we are defining as our super enhancers in these different cell types. The most interesting thing that we found about CD4-CTLs is that if you look at any lineage of T helper cells, they are slightly demethylated compared to naive cells. But when you look at CD4-CTLs, we're now comparing the degree of demethylation. So here are our two controls. This is showing you one example here. This is the degree of methylation, whereas the CD4-CTL is dramatically reduced. It goes down to about 65% of methyl CpG compared to naive. Methylation usually is the simplistic way to say it is if the gene is highly methylated, it's not going to be expressed. If it's demethylated, it's going to be expressed. So just to show you a couple of drivers. So RUNX3, important transcription factor for CD4-CTLs. You can see these two lanes are showing you CD4-CTLs. You can already see open chromatin here, which is not seen in naive cells. Let's show you one more example. If you think of perforin, crucial in CD4-CTLs and CD8 cells, open chromatin. So you had to demethylate heavily during development. You had to take a CD4 cell, it went through a program, you demethylated it heavily in order to now get the expression of this new cassette of genes, which are making it more like a CD8 cell. This is the naive cell, this is the CD4-CTL. Obviously, I just showed you a couple of examples. The other question you should ask is, did therapy lead to attenuation of CD4-CTLs? So, if you got the disease to improve, do CD4-CTLs decline? And so in IgG4-related disease, we had done this. So here we treat patients with rituximab. This is just depleting B cells. And you can see clinical outcome improves dramatically with treatment. IgG4 levels don't drop that much. They drop a bit. B cell depletion is perfect. You can expect that. Plasmablasts do not have CD20. So rituximab targets CD20, but they decline after time because they have to come from B cells. They have to be B cells have to be activated. So if you look after a month, they've started to decline quite a bit. CD4-CTL, the effector memories drop. CD4-CTL clones, we can follow specific clones because we know the V beta sequence, they also decline. Okay, so there is a decline in the CD4-CTLs when treatment works and patients get better. So this has led us, and I'll only touch on this again to think about methodology. How do you approach things? So B cells, what role are they playing in fibrosis? If you depleted B cells, disease got better. Is there anything they do beyond presenting antigen to a CD4-CTL? So we have I just show you this to show you an example, just using t-SNE plots. And this is we do 25 color flow, but this is using a few colors to compare and paint the cells. If you look at the control, and then you look at somebody with IgG4-related disease, and this is from about 40 patients, we've got similar results. You can see that there are some new islands over here, which did not exist in the control. You can see the other islands are virtually identical. Okay, everything looks similar. You just look at the patterns, they're almost identical. But these islands here, these ones with arrows, do not exist in the control. So novel cells are being created in a disease context. We now understand that all of these cells are created outside the germinal center in an extrafollicular site. And I'll show you some of that data in a COVID context tomorrow. These cells, some of them are secreting lysyl oxidase 2. Okay, and lysyl oxidase 2 can cross-link collagen. This is to show you that, yes, in the disease tissue, we have cells that are making IgG4 and are also making lysyl oxidase 2, and that this is something that goes on. Can we prove it in a human that is important? No, it's hard to prove. We can only quantitate it. But so that's why we always need to go back to animal models to prove mechanism, which is something that we cannot do just by looking at humans. Okay? So again, the big picture view, we come back to this. This is the pathogens of autoimmune fibrotic diseases. You have CD4-CTLs, you have activated B cells, they may work together, and you get cell death. Okay? So I'll spend a few minutes talking about antigen discovery. Obviously, you want to know what antigen are the activated B cells seeing and what antigen activated T cells seeing. Are they the same antigens? And T-cell antigen discovery is more difficult, but we are doing it now. I'll mention it at the end. If you're interested, I do have some slides on how it's done. So for B-cell antigen discovery, pretty easy. B-cell antigen discovery, what we do is we make monoclonal antibodies. And this is what's same thing was done by the way in COVID, the similar approach, what I'm showing you here. So you take a patient, you take the plasmablasts, he's got active disease, you put one cell per well. Okay, and then you from each cell, you pull out the antibody heavy chain and light chain gene. Okay, then you put these into cassettes where you can have the rest of the antibody heavy chain and light chain. You PCR it out, put it into a cassette, and you make a monoclonal antibody. It's a human monoclonal antibody, one coming from one from each cell. And this is what is done. But the antibodies that Trump received, I can't dignify him in many ways, but he basically received 2 monoclonal antibodies derived in this fashion from a COVID-19 patient. Okay, well characterized. Anyway, so these antibodies in this case were staining some cells. It was interesting. This is showing you that by next-gen sequencing, these peaks came out to be the same as what we picked by single cell. So the ones that are more commonly as single cells were also more common by next-gen sequencing because can actually sample more cells. And then we stained pancreatic cells and shown that, yes, these antibodies are staining some antigen in pancreatic cells. So then it's easy to try to purify it. Fairly simple to take the antibody, conjugate it to cephros, make a lysate of pancreatic cells as a source of antigen, do affinity chromatography, do mass spec. And this is what the mass spec shows us. There's always some noise, but you can see in all 3 clones that we pulled, the peak antigen picked up was galectin-3. So we now know in a subset, it's only about 1/4 of patients or 1/3 of patients with IgG4-related disease, galectin-3 is an antigen. In some other patients, we did with Bill Robinson's lab, we used microarrays looking for anti-cytokine antibodies. He found a very interesting anti-cytokine antibody, which is against the IL-1 receptor antagonist. Might explain why the disease gets worse because IL-1 is being made by CD4 CTLs as well. So that's something else, another approach that we've used. There are other approaches like Plato that we've used and so on. And so there are multiple approaches that can be used for B-cell antigen discovery. For T-cell antigen discovery, which is what we're doing right now with Michael Birnbaum, who's our neighbor, he's at MIT actually, but he's next door. We are the Reagan Institute is next to MIT actually. So we take, in this, what you do is, so single cell RNA sequencing, and we do TCR sequencing, so on those single cells, and identify clonal expansions, and then we make soluble T-cell receptors. So you made the T-cell receptor from the biggest clones. But you predict which is the relevant HLA by clustering patients. So let's say I have 50 patients, and 10 of them had the same T-cell receptor, V beta usage, or V beta plus J beta usage, maybe the same TCR alpha usage. So they have very similar T-cell receptors. Those who share T-cell receptors must share HLA. I mean, that's the assumption we make. And this method was really derived by Mark Davis called GLYPH. So we worked with Mark Davis on this. So then you try to predict the relevant HLA by doing this. This is actually done quite successfully by us. I don't believe it will work every time. It works sometimes. So this works, at least in this disease. Then you take, this is again done by Michael Birnbaum, you take the library of all human peptides, all known human peptides, and attach it to the HLA of choice that you discovered by GLYPH. And then you screen a library and ask, the soluble TCR from that big clone, does it recognize, which peptide does it recognize, and thus you identify the peptide. Now, I do have slides. If you're very interested, I'll go to those slides, but I'm going to close with a summary of an overview of immunology, but it'll be in a different sort of presentation form. I just want to mention that a lot of people in my lab were involved in this work. Almost all of them are involved in various aspects of our work, whether it is on systemic sclerosis or COVID-19 or IgG4-related disease or on the single gene mutations. There are some ex-members. There's actually one person who was at NII many years ago who was a key person in my lab. There's Hamid Mattu. Vinay Mahajan is actually a graduate of All India Institute of Medical Sciences, and did his PhD at MIT after finishing medical school there. And there are others who are very important. I have key collaborators. John Stone is my key collaborator at MGH. For all of us, we do clinical studies, clinical trials. I mean, I can easily convince him to do a clinical trial. There are many fellows in my lab who are shared between who actually go to the clinic and also come and work in the lab. Seiji Nakamura has been helpful in Japan with a lot of clinical material. Dinesh Khanna is my key collaborator in systemic sclerosis. He's at the University of Michigan. For those of you who are interested in Bollywood, he's closely connected to an ex-Bollywood person. I won't name him right here. And there are other people who are involved. Ramnik Xavier for microbiome, I didn't talk about it. Alex Shelik has helped us a lot with single-cell RNA-seq. And then I didn't talk about a single gene mutation project where we have other collaborators. So I'm going to end with the lymphocyte wrap. I think I'll stop sharing and I'll do this.
Yeah.
And this is just to summarize the whole, approaches to immunology. I have to warn you that unfortunately, this involves my moving my hips. You are not going to be able to see that. My hips are moving. I'm sitting on a chair. Just know that my hips are moving while I do this.
Thank you.
Pathogens lurking everywhere.
Yeah.
Go get them first. They never fight fair. Give me a D, give me a J, put a V in frame. It'll be A-OK. Surrogate light chains, pre-TCR, kappa and alpha can't be too far. Thread that peptide into tap. Everybody do the lymphocyte wrap. Naive cells dance from hall to hall, pining and hoping to get that call, looking for antigen below and above. Many will die of unrequited love. Thread that peptide into tap. Everybody do the lymphocyte wrap. DC's lurking everywhere. They'll get your antigen, don't despair. CCL21 to CCR7. Come in, DC baby, make this lymphocyte heaven. Thread that peptide into tap. Everybody do the lymphocyte wrap. Wake up, T cell, activate zap. T cell says, takes 2 hands to clap. Give me signal 1, give me signal 2, and I'll give you a cytokine brew. Thread that peptide into tap. Everybody do the lymphocyte wrap. Collectins, complement, macrophages. That's what you get in the early stages. In antibodies, cytokines, CTLs too. Hey, pathogen, that's the end of you. Thread that peptide into tap. Everybody do the lymphocyte wrap. Thank you for listening. I'm done. I can take questions.
Thank you very much on this poetic note. So we have some wonderful, very, very vast canvas you have covered, Shiv, and try to make it understandable to all of us. And I have some good questions here. This one first is from Amita Agarwal. So she is asking, is there anything similar between the antigens in histoplasma capsulatum and endothelial cells? Could it be that SSC, the CD4 CTLs are recognizing some microbial antigens like CMV, which can stay in endothelial cells?
Okay, can you still hear me?
Yeah, we can still hear you.
Okay, so I actually don't think there's any connection because I don't think there's even any clonal connections. We have done repertoire on the fibrosing mediastinus patients, T-cell repertoire. These all patients that came from Vanderbilt to us. We have done repertoire on patients with IgG4 related disease. We have done repertoire on a lot of patients with scleroderma. There's no commonality between the diseases. I do think these are very distinct antigens. Okay. And the CD4 CTLs evolved to deal with pathogens. We just happen to use it, unfortunately, in disease.
Okay, so then there is another question. So, sir, you made a statement, break in tolerance is one of the reasons for disease. Does it mean that we have now started focusing more on danger hypothesis over horror autotoxicity as a reason for induction of immunity?
So Paul Ehrlich used the term horror autotoxicus to refer to the fact that, the immune system will probably attack us And that is the thinking about tolerance and autoimmunity. The earliest person to think about it. He also thought about tumor immunology and many other things. So the danger hypothesis is a little different. Danger hypothesis is what we attribute is to Pauly Matzinger and a little bit to Charlie Janeway, which really explains the need for the second signal, that you need to have an adjuvant to induce B7 or some such thing. That the immunologist's dirty little secret, as Janeway called it, was the adjuvant. So that's a different thing. There is no immune response that you get without a danger signal. Whether it comes from DAMPs or PAMPs, there's always going to be some danger signal to induce it. In every disease, I mean, in an autoimmune disease, it's coming from some cause of activating the immune system. The dendritic cell has to be made to make B7, and that always must happen. Now, obviously, in a pathogen, it's easy to explain, and in an autoimmune disease, you have to think of other explanations, but they exist. So, yes, we think break intolerance actually is common even in infectious disease. Some of you may know the report about COVID-19, where like almost 10% of all patients have autoantibodies to type 1 interferons. This is from John Laurent Casanova. So yes, break intolerance is pretty common. And I think in inflammatory disease, it's pretty important.
So what's the longevity of CD4 positive CTLs in vivo?
So we don't know the answer in humans for CD4 CTL longevity because they're probably constantly being produced. We presume it's not very long-lived because, you know, when you treat patients and you treat them with a B-cell depleting agent, in a matter of weeks, the CD4 CTLs are lost. So actually, I presume that many effectors in every type of effector is actually not that long-lived and requires constant replenishment by activation of precursors.
Could these cells be responsible for excessive damage of endothelial cells and consequent fibrosis of lungs in COVID-19?
So in theory, that could be, but I can already tell you, no, we looked. I actually will tell you that in COVID-19, I'm spilling the beans here, that paper, this is our second story on COVID-19. There's no good evidence for T cells doing things, especially early in disease. If you look in people who have died within a week of symptoms starting, and you look at the lungs, and we have looked at more than 20 people's lungs, there is no evidence for T cells doing a lot, and not for CD4 CTLs. So that was a good thought, but that's not what's happening.
All right, any question from panelists, please?
Yeah, Pawan, I would like to ask a small question, if you allow me. Sunil here.
Yeah,
Yes.
Go ahead.
Okay, Shiv, you say that the CD4 cells, they don't do anything in the COVID, but what is this cytokine storm coming from then?
So I may summarize this for you tomorrow as well. So if you look in the first week, so we are not the only people to look at lungs. So if you look in the first week, you can look at the first week and you look after 3 weeks. First of all, even the guys who are very severely ill, and you're looking at people who from day of symptom onset to unfortunately succumbing to disease is under 8 days. There is, first of all, severe lymphopenia. CD8 T cells in the lymph node, we are looking at draining lymph node and the lung together. CD8 T cell in the draining lymph node is going through severe activation-induced cell death. Okay, there's very little CD8. The CD8s are not expanding. There's some Th1 expansion, but not seen in the lung. Very little seen in the lung. So I think most of the disease, if you think about COVID-19, initially, I mean, they're not saying there's no role for T cell ever. Initially, the disease is really driven, I think, by severe innate immunity because the virus is so good at evading type 1 interferon induction, you know, and I'll talk more about it tomorrow. The virus in the susceptible person is multiplying rapidly. DAMPs, there are DAMPs created, and the PAMPs include spike. I think there's a huge role. There's a recent paper out. Huge role for alternative pathway complement activation via spike. There's a report that's on bioarchives but has not yet been accepted anywhere, which therefore I'm not taking seriously yet, showing that the N protein also is activating the lectin pathway of complement. So I think a lot of the damage is coming from viral infection, cell death, activation of inflammation, lot of myeloid cells pouring into the lung. You have mainly, what do you see there? You see lots of nets and you see lots of macrophages. You see very few T cells. I'm describing all the early stage patients. And in the paper I'll talk about tomorrow, we have looked at early stage and late stage separately, but that's all lymph node, right? Later on, there are some T cells. Later on, there's some recovery of adaptive immunity. The lymphopenia decrease, but that's like 6 weeks into the disease. So I actually don't think, while there might be roles for T cells in giving you some protection and so on, which is all hard to prove, I don't think there's a good rule or any evidence to say that T cell is driving the disease, and it's not innate immunity and not complement and not neutrophils and not macrophages. So I think we have to be cautious.
Thank you.
Shiv, can I ask you a question? This is Joshi.
Hi, Joshi.
Yeah.
Hi. So do you think that these vaccines based on spike proteins will be successful? What is your read on it?
So, so Joshi, I should say that, so even with some adjuvants, although you do get a little bit of TNF induction, for the most part, so as I'll talk tomorrow, you won't be there tomorrow, but for the most part, so what we just published a month ago, for the most part, germinal center loss is because we think because of TNF induction in the lymph node, blocking TFH differentiation into GCTFH. And that's why we are seeing the striking loss of germinal center. Towards very late in disease, just before some people have passed away, unfortunately, there's some little bit of recovery of BCL6 positive cells. So I think, and there's some data now from Patrick Wilson, which you'll see in some time. So I think when patients actually don't die and they recover, right? They got very sick and they recovered, then they start to make, during convalescence, they start to make some germinal center response. So I actually think that if you immunize with spike, it's not the virus, screwing things up. It is basically just spike and some adjuvant, hopefully not like a potent adjuvant that is going to destroy the immune system. It should be fine. And you should make antibodies. Now, we still haven't seen durability, but I have seen data on durability on the, so the Johnson & Johnson is Dan Brook, who's my colleague at the Reagan. He's the one who's doing the Johnson & Johnson vaccine, which basically his lab made. But he has used that vaccine in Zika. He's used that vaccine for RSV, the same platform, used the vaccine for HIV. And there is now evidence in those situations that you have immunity for 2.5 years, nothing more than 2.5 years. So I'm hoping that some of these vaccines will give you reasonably durable immunity, and that should be enough for us to figure out, and that it will work. And I actually think it will work really well because, as you know, the spike, you can't afford to mutate the RBD yet. So we will not have a major issue. First of all, it doesn't look like there'll be any enhancement. Secondly, it's not like HCV or HIV, where you have so much mutation that you can't recognize anymore.
All right, there are some questions, but they are all related to COVID-19. I think we have to wrap up this lecture. Maybe tomorrow you will be talking about and
yes,
I will get those answers. Okay. So thank you very much, Shiv, for a very entertaining and very informative, for me, very mesmerizing talk. Thank you very much.
And I have to say hi to Joshi and thank you all. Okay.
Okay, so we move on to the next item on the agenda. And it gives me great pleasure to welcome Dr. Joshi Jacob in this evening here, morning there. And he will be the second speaker on the keynote session here. And he'll be speaking on B-cell responses and establishment of humoral immunity. Dr. Jacob grew up in southern India, where he did his primary education and moved to the United States for higher education. He earned his PhD in immunology from the University of Maryland School of Medicine in Baltimore and did his postdoctoral training at the Rockefeller University and the Massachusetts Institutes of Technology. Currently, he's the tenured professor at the Emory University School of Medicine in Atlanta, Georgia. His current research interests focus on developing novel antivirals against SARS, coronavirus 2, dengue, and influenza viruses. But I believe these are his seasonal interests. but I think his abiding interest has been in the understanding antibody responses against variant influenza viruses. I believe all this stems from Professor Jacobs' abiding interest in understanding the cellular and molecular basis of immunological memory, how B cell memory is generated, regulated, and maintained. I'm reminded of his seminal work on CD28 B7 interactions in regulating the plasma cell function. His studies aimed at unraveling various shades of phenomenon of original antigenic sin and so on, all directed towards understanding long-term memory of protective immunity, so very important for designing effective vaccines. Coming to his keynote address today, we all know about remarkable ability of the immune system to respond to a seemingly endless array of antigens and to respond with enhanced vigor to pathogens encountered in the past. Again, the question of memory here. So Professor Jacob will present today an in-depth exposition of the events that follow antigen activation of B cells, and this will include differentiation into plasma cells and memory B cells, class switching and affinity maturation by somatic hypermutation and antigen-driven selection, and establishment of B cell memory. It's my pleasure to invite Professor Joshi Jacob to deliver his keynote address. Dr. Joshi Jacob.
Thank you, Pavan. That was such an awesome introduction. For a minute, I thought it was like my retirement party or something. It is always difficult to do two things: to be the last speaker of the day, and also even more difficult is to follow Shiv, because to perform like him is almost impossible. So what I have done today is not the current projects that are ongoing in my lab, but if you want to know, I'm happy to tell you. But what I'd rather do is it's like a lecture on B cell activation, the things that happen to a B cell and how you make memory. So if you haven't had a class on this would be like a good lecture for you. And I should be done in like 35 minutes. I've timed it. One moment. Okay, so this slide tells you the success of vaccines. So if you look at this slide, it tells you that following vaccination, when we got to 1980, we eradicated smallpox because you can actually immunize and then get rid of the infection. And this is again to show you three different diseases: diphtheria, polio, and measles. And it shows that when you introduce the vaccine, there is a precipitous drop in the incidence of infections, whether it be diphtheria or polio, and even measles. And in measles, you get it as a child, and then you can get it again, and it comes back in SSPE. And then what this shows is once the vaccine was introduced, the incidence of this SSPE also goes down precipitously. Okay, and so you want to get vaccinated, and you do not wish to be an anti-vaccine. Okay, so what is the purpose of vaccination? It does three things. One, it induces memory B cells so that you have an army of cells that can recognize the pathogen that you've seen before. You have memory T cells, CD4 and CD8 T cells, that can recognize previous insults. And also you have helper T cells that will help memory B cells to give you a profound immune response when the antigen comes the second time. And better yet, you have this creation of plasma cells that can produce antibodies for a lifetime, and they reside in your bone marrow. So if I had chickenpox when I was 5 years old, and I'm 50 years old, for the last 45 years, I've been making a lot of these antibodies because I have these plasma cells that continuously make these antibodies. Okay. Right. So now, once you have vaccinated, what happens when you get exposed to the pathogen again? Step number one, you have circulating antibodies that will form immune complex with the pathogen. You activate the complement pathway. You have inflammation. You have activation of memory CD4, CD8 T cells. You have enhanced killing of the virus-infected target cells. You have activation of memory B cells and the rapid differentiation to plasma cells. And you have massive antibody production. And for the most part, you do not even know that you got a secondary exposure because the system is so efficient at dealing with repeat responses to things seen in the past or things that you have been vaccinated to. Okay? And finally, what that does is the pathogen has no chance. It is decimated. So what the pathogens do is to escape this, they mutate away and they change, and that becomes a challenge. Like in flu, that you vaccinate, then you control the virus, and then it mutates, and then you make another vaccine and so forth every year. So this is an introductory slide. There are 3 stages to host defense. The first is innate defense. Innate defense is like the barriers that you have, like the Great Wall of China, the barbed wire, the things that separate you. And then you have adaptive immunity, T cell and B cell response. And finally, you remember things. It's like the serial, the soap operas you find on television. The Sas and the Bahu stories. They always remember everything. They have incredible memory of all the past insults. And so this shows you a duration of infection and the level of microorganism. So the level of organism, it comes in, it divides, it gets to a high number, and then you need to knock that down to a threshold level by the innate immune responses. Only then can your adaptive immune system start, okay? And if you are missing the innate immune response, then if you see this line that goes up, the pathogen number goes up, and through the roof, and then you cannot get over it, and then you most of the time you die. I have to tell you, all of the adaptive immune system comes into play, and it's crucial for sure. But if you have no innate immunity, you could be dead as a doornail. So let's move to B cells. So you have seen this 1,000 times. I'll just go through it very quickly. You have a large repertoire of B cells, and each B cell encodes a single specificity for the most part. Okay? There could be some level of promiscuity, but for this lecture, let's just assume that it's monospecific. That's because it has only one specific rearrangement in the like Shiv referred to. And so when antigen comes in, it picks out from this large lineup of B cells those that are specific, and they are induced to proliferate. And then you have all the subsequent reactions that go on that I will talk to you in the next 20 minutes or so. Okay. So we need to define a few things. We need to talk about what antigens are, especially what an antigenic determinant or an epitope is. And then we'll talk about affinity. We'll refer antibody, we'll refer to affinity and avidity. So shown on here is, this is the antibody combining site. This is the part of the antibody that binds to the antigen, shown on this blue. And the antibody does not swallow the whole antibody. It only sees a small piece of it, okay? And that small piece that it sees, it's called an antigenic determinant or an epitope. And so what happens is you need to have a perfect fit in order for it to bind really well. So this is a good fit, and the antigen-antibody complex stays bound. And on the right-hand side, this is a poor fit. This antibody can bind to it, but it is not perfect in its binding. So what happens? This is a low affinity interactions. And the reason why you need an excellent fit is because the interactions that hold the antigen-antibody complex together are actually very weak interactions, including hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals forces. So what that means is these interactions operate over a very small distance. And so you need to have like a lot of these together in order to hold it tight. It's like a Velcro. Having one of them is not good enough. When you have many of them, it's almost impossible to pull them apart. It's the same thing with antibodies. Okay? And this is just to tell you that you can actually measure the strength of this binding in terms of affinity and also avidity. And people tend to use this interchangeably, but it is not. Affinity just tells you one combining site and one epitope. The strength of binding, that is the affinity. And the avidity is the sum of all the if you have like an IgM molecule that has like 10 combining sites, if you added all of them, you would call that the avidity, right? Very good. So the antibody avidity is based upon the isotype, because you can have either 2 combining sites or 10. And so the avidity is the total strength of interaction between a multivalent antigen and the antibody. Okay? Right. So I'll just summarize the effective functions of antibodies in just one slide. So B cells get activated, you make antibodies. These antibodies can neutralize microbes and toxins. They can opsonize and phagocytose microbes. Okay? So they bind to the microbe, and the FC receptor of this antibody that's bound to the microbe can bind to an FC receptor on a phagocyte and can be taken in easily. And also, like Shiv referred to, this antigen antibody complexes, once formed, they can activate the classical pathway of complement, and which leads to lysis of the microbe, phagocytosis, and inflammation. Okay. All right, very good. So cross-reactivity, and in the first slide I showed you that the B cells are very specific, but however, what happens is, even though they are specific, the antibodies can cross-react with other related antigens. So what happens is, when two different antigens share a similar epitope, and this antibody cannot discriminate between the two, okay? So when you make antibodies, and what the bug does is the bug keeps changing itself. And it wants to have the epitope look like some part of your body, so that the target for this antibody is much more to attack your organs in your body than it is to attack the microbes. And here are a few examples. So molecular mimicry is when the epitopes between the pathogen and the humans look very similar, and the antibodies made against the pathogen can cross-react with the self-antigens and cause disease. And this is just there are three categories. So the exogenous antigen, Streptococcus M protein, it looks like cardiac myosin. So when a child gets infected with Streptococcus, you make the antibody, and there's much more cardiac myosin than there are bacteria, and then the antigen will attack the cardiac myosin and cause rheumatic fever. Similar, Yersinia protein can cause Graves' disease by cross-reactivity. Papilloma E2 looks very similar to insulin receptor. Measles can lead to EAE. MTB can cause rheumatoid arthritis. Poliovirus VP2 looks like ACH receptor, and antibodies can cause myasthenia gravis. And the bacterial phospholipid looks like DNA and can cause SLE. Okay, good. So two things to remember: polyclonal versus monoclonal. Polyclonal means that I told you one B cell can make one specificity. It attaches to one antigenic determinant. But as shown here, it's a bacterium. It has many different epitopes shown by all these sizes, and you can make antibodies against all of these different epitopes. So the sum, all of these antibodies, that'll be a polyclonal response. But if you can isolate just this one clone that makes antibodies against this one determinant, that is called a monoclonal. And the people that would discover this would go on to win the Nobel Prize. Okay, why is this important? This is important because this has actually changed the way treatments have evolved. It started out with murine monoclonal antibodies. You would make monoclonal antibodies, inject into humans, and you could cure diseases. But the problem was, you would make a human anti-murine antibody response, and that was not good. Then what they made chimeric antibodies, where 30% of the chimera is murine and 70% is human. That is actually pretty good, but nonetheless, it's still, you can trace antibodies against the 30%. And then came the humanized antibody, where they take the murine or rodent antibody, they keep everything except the antigen contacting sites. And so the CDRs are rodent, everything else is human, and that was great. That worked beautifully. And the last type, what we do now, is human monoclonal antibody. So you just sort out the B cells, clone out the heavy and light chain, you can make the antibodies, and then you are in business. Okay. So there are four types of therapeutic monoclonal antibody. One is the mouse, pure mouse, and you can tell it by the way that it is named. The name ends in MOMAB, so it's a mouse antibody. Chimera would be CMAB, like rituximab. And then if it's humanized, it's actually a rodent one, but the rodent part is just the top part shown in blue. It's a Zumab. And if it's completely human antibody, it's called a MOMAB. So next time you find an advertisement on television or on a journal, you can actually tell what kind of antibody is based upon the last, how the name actually ends. Okay. So I'll just give you like two examples, and then I'm going to move on. So rituximab is a monoclonal antibody directed against CD20 on B cells. It's beautiful because if you give it, it can actually deplete B cells. And this is a patient that came into the clinic at Emory, and he has pemphigus, and which he's an autoantibody disease. And they bring him in, and then they treat him for 2 months versus 4 months with the rituximab, and then lo and behold, in 2 months, all his lesions heal up because it kills it depletes the B cells, and then it's completely cured. Okay. And if you look at turn on television in the United States, you see this commercial for Humira all the time. It is actually a recombinant IgG1 monoclonal antibody against TNF alpha. This is actually the top-selling drug in the United States today, about $17 billion every year. And if you look at what is the drug that makes the most money in the US, right? The 7 out of 10 are actually antibodies. So the point I'm trying to make is the applications of this are actually super, super powerful. So there are two types of active immunity versus passive immunity. So in active immunity, you induce immunity in the host by activating the immune system and generating immune memory. You do that by either getting infected or by vaccinating the person. Okay, and usually you have to vaccinate before the anticipated exposure. Right? If I'm going from here to, let's say, Africa, and I'm worried about yellow fever, I should get the vaccine now, wait a month or so to get the antibody responses going and the T-cell response, and then go to the spot, right? The exception is rabies. You can actually wait for the dog to bite you, and then you can actually go get the vaccine. And that is because the incubation period of rabies is much longer than it takes for your immune response to develop. So that's okay. You don't have to, everybody go get a rabies vaccine and hopefully you get bitten. You don't have to do that. You can actually do it post-exposure. Okay? And passive immunity is the transfer, and we hear this a lot now. It is the transfer of convalescent sera monoclonal antibodies that you put into a patient, and then you can actually cure them by doing this. Convalescent sera used to be what they used to in the old days, but now we move on to monoclonal antibodies. And these things are actually very efficient. So in 1988, serum from horses were used to immunize. Serum from horses immunized with diphtheria toxin were used to treat patients. Okay, 1890, we developed tetanus anti-serum. But the problem is you can only do this once, because if you do this again, then you make an anti-horse response, and then you know what happens next. And if you got botulism today, you still get a horse antitoxin, and hopefully you don't get botulism a second time. And we already know from the Ebola outbreak and also this current COVID-19, you can actually transfer convalescent plasma, and then you can actually contain or control the infection. And I have to remind you, during the outbreak of Ebola, this is my university, Emory University School of Medicine in Atlanta, and this is the missionary doctor that got infected with Ebola, and he came to the U.S. and they gave him a monoclonal antibody. I'll show you in the next slide. And he's cured, and he's leaving the hospital. He's very happy. And so what they gave him was ZMapp. They were 3 monoclonal antibody cocktail that they made in transgenic tobacco plants. And they give him the antibody, and then they can cure him. The thing you have to remember is you need to put it's not a simple exercise. You had to put a lot of protein into a person. So the scale or the magnitude of antibodies that you require is actually quite high. And so then this is a doctor that got cured, and this was a nurse in Dallas that got infected. So he cured her by giving his convalescent plasma into her, and she gets cured. And this is the gentleman that died, and he's from Liberia. He died in Dallas, and you could not give a plasma transfer to him because there was a blood group mismatch. So you have to give plasma only if you're of the same blood type, and no incompatibilities. This one worked out, this one did not work out because of the incompatibility. So that's why now we make monoclonal antibodies, and that you can give it to anybody, and it works really well. And this is in the news. Regeneron is a company in the States. They make these antibody against the SARS coronavirus, against the spike protein. And you can actually give the antibody, and the hope is that you can actually cure people. And they actually gave it to our fearless leader, and this is the antibody that the President Trump received like last week. And the outcome, it's not published yet, but the hope is that it's excellent therapy should have taken care of his viremia, at least to some extent. So now we have talked about, just to recap, antibodies are important. they are important for serological memory. I talked about one lymphocyte, one specificity. Then we talked about monoclonal antibodies, the applications, why this is so important. And now we'll just get into nuts and bolts of what happens when a B cell gets activated, and what do you do? How do they go on to establish memory? And this is not, and this topic, when we teach it at Emory, this is like a 3-week class where we go into each individual thing. But this is a quick review of what happens. It's like a 300-feet view of it. Okay. So antigens comes in two flavors: T independent, which, as the name suggests, you can activate a B cell without T cell help, and obviously you have T dependent antigens which require T cell help. And in a T independent antigen, you have the B cell receptor, B cell, and you have B cell receptor. And what happens is the antigen can activate the B cells in the absence of T cell help and make them differentiate into short-lived plasma cell. And when you do this, what happens is that there is no immune memory for the most part. You need T cell help in order to establish immune memory. So when your nephew or niece or your child gets repeated ear infections with bacteria, that's because it's a T-independent response. And so they can infect you again and again and again and again. If you have a T-dependent response, you will have this part of the response, plus you'll open up and have a much more elaborate immune response. So if it's a T-dependent antigen, you do make the short-lived plasma cell response. And you also make a T-dependent germinal center B cell response. And in the germinal center B cell response, this B cell undergoes mutation and selection. And then the B cells that can have the most high affinity or better binding antibodies survive. The rest of them perish. And those with the jackpot mutations or high affinity go on to become plasmablasts. They go to the bone marrow and they live in the bone marrow for extended periods of time, and they make antibodies for their life. They can make between 10 to the 4th and 10 to the 5th antibody molecules per second. So it's a factor. You don't need a whole lot of these cells to make a lot of antibody. And I have to tell you, based upon the isotype, if it's IgG, 2A or something, you have 21-day half-life. So it's very easy that you make a high amount of antibodies in your body. And from the germinal center also comes memory B cells, high affinity memory B cells that stay and they wait for the antigen to come again so that they can actually differentiate faster and go on and become plasma cell, make more antibodies. Okay. So T-dependent antigens, these are the characteristic T-independent antigens that are type 1 and type 2. And I just put the slide. You can look at it at your leisure. I'm going to skip in the interest of time. So there are two events that happen during a B cell differentiation. One is class switch recombination, and the other is affinity maturation. Okay. Class switch recombination. So this is a antibody. Everybody starts off as IgM, and when this B cell gets activated, it keeps the specificity the same, the front end the same, and it switches out the rear end for another heavy chain isotype. So you go from IgM to IgG. The front end remains the same. The back end changes. It's like one of these, 18-wheeler trucks that you see on the highway. So the front end is always the same. And today they will be carrying, produce for a grocery store, and tomorrow they could be carrying, gasoline for Indian Oil or something like that. So the front end is the same, and the back is the one that you keep changing. And the reason why you do this is because you can actually fundamentally change the effective function of antibodies by doing this. Just to quickly, IgG, D, and E have 2 combining sites. IgA has 4, IgM has 10. That's one thing. So the avidity, like I told you, avidity would be so if it's the same combining site, right? The affinity of the IgG and the IgM is exactly the same because it's one combining site and one antigenic determinant. But if you look at avidity, this is 2 and this is 10. So you have 5 times more combining sites. So the binding efficiency, avidity, is significantly higher because of the valency of this antibody molecule. And the reason why you switch between these is, so this is the rearranged VDJ, this is the C mu, and all the downstream switch heavy chain isotypes. So what happens is you have these switch regions, S mu, and other upstream of each of these heavy chain isotopes, you have a switch region, and you switch between them. You bring them together, you cut it, and you glue it together, and now you went from an IgM to IgG3C gamma 3. If you switch all the way to the end, C alpha, now you have an IgA antibodies. And once you recombine it, you cannot say, oh, I just changed my mind. I want to go back to be an IgM. That is impossible because you have actually deleted the intervening DNA at the genome level. So what controls which way the B cell decides to switch? And it turns out it has to do with the cytokine that's produced. So the T cells that make the cytokine, during the immune response, the cytokines that are produced will actually determine what isotype of B cell switches to. If you make a lot of IL-4, go to IgG1 or IgE. If you make a lot of TGF-beta, which is what you find in the mucosa and in the gut, you have a lot of IgA that is induced. And depending upon the isotype that you switch to, you have a lot of these different functions that it's the same antibody, but by changing the rear end, you have completely changed the way that the effector mechanisms that are involved in this antibody. I just show you just a couple. You don't need to go. You have seen this before if you have taken a class. If you look at complement activation, there are some that do, others that don't. If you look at placental transfer, there's some that do, most of them that don't. If you look at the binding of macrophages and phagocytes, some do, the others don't. If you look at binding to mast cells and causing degranulation, all the IgE do, and so forth. Okay, so there is a consequence to switching your heavy chain isotype. But the most important thing is the isotype changes. If you want to make a vaccine and you want it to be a long-lived antibody response, you want to hit it with an adjuvant. You put the right kind of adjuvant that makes the right kind of cytokine that forces the B cell to switch to the isotype of your choice. So you want to hit something that goes at 20 or 21 days, as opposed to something that goes like 3 days or something. You want to stay away from things that you vaccinate and you make an IG response because you don't want to do that at all. It's not going to end well. All right. So now we'll get into mechanisms of, how do you bring about these class switch recombination. Okay, so this is Dr. Honcho. He won the Nobel Prize. He discovered an enzyme called activation-induced deaminase (AID), and this enzyme is important because it induces class switching and immunoglobulin somatic hypermutation, which I'll tell you in a minute. Okay. All right. So in class switching, what happens is again, so this is the VDJ, and you have the switch sites ahead of everything. So what the AID does, it makes a cut, and then you glue the cuts together by non-homologous end joining. So you have AID mediated double-stranded break. You break it, and then you have end joining, and then and that's how you bring these two pieces together, and the intervening piece gets thrown out. Okay. This is just to show you that it's the same slide as before. You have VDJ, C mu, and C epsilon, and you have AID that cuts, and then you glue it together, and now you have a VDJ C epsilon. You made an IgE antibody. All right. So B cell differentiation. So a naive B cell gets activated. You have membrane-bound IgG and becomes plasma cell. It secretes the immunoglobulin, and the way that you go from membrane-bound to secreted, you do it by differential splicing, and everybody knows that. I just put it for the sake of completion. And the other thing that happens is affinity maturation. So when you start out, the affinity is kind of low. With time, you make better and better higher affinity antibodies, and this progressive increase in affinity or affinity maturation is caused by two events. One, you mutate, and then you select for the high affinity clones. Okay, all right. So this is a busy slide, but it's actually very not that difficult. So this just shows you day 7 of an immune response, day 14, and day 21. This is heavy chain and light chain. Let's not worry about the light chain. Let's just do heavy chain. If you look at mutations, you find that there are mutations that are introduced into the heavy chain. And what happens is with time, the mutations get selected in the CDR regions. And that CDRs are the ones that actually contact the antigen. And by selecting for mutations that are in the CDRs, you start out like it's a poor fit. It binds the combining site, that's the epitope. Then you mutate, and then now it becomes a perfect binding and a high affinity binding, and then that is very stable. So there are two things. You make a lot of these mutants, and then you select for the ones that actually bind the best. And the rest of them that do not bind are not enticed to enter into the next level of selection. Okay? So antigen-driven B-cell differentiation. So this is just a stepwise progression of the things that happen. Day 0, antigen comes in, the DC is taken. So again, this is the T cell zone, B cell zone. This is the interfollicular region. So the dendritic cells take up the antigen, they present it, and those T cells that can see the MHC plus antigen will come and get activated. Similarly, the antigen in the B cell zone, the B cells that are specific for the antigen will bind to it, and then they get activated. And once they get activated, they leave their homes, right, from the T cell follicle and from the B cell follicle. They come outside, and then the B and the T cell stay glued to each other, and it's almost like they are kissing, and they do that for almost 2 days, right? It's almost like a newly married Indian couple on their honeymoon. And then what happens is, on day 4, 5, 6, and 7, they come back into the B cell zone, both the B and the T cells, and now they start expanding and they push everything out. And then now you have what's called a germinal center that is formed. And it is in this germinal center that you have this affinity maturation that happens. Okay? All right. So this is a germinal center, like I showed in the previous slide, that you have antigen that's bound to the follicular dendritic cells. And first is seeding. The B cells and B and T cells enter. In the second, you have proliferation and somatic mutation in the dark zone. And then they move up into the light zone, where they are selected for by binding to the antigen. If the B cell mutated away and cannot bind the antigen again, then it stops there. It cannot go forward, because you need antigen binding to rescue these cells. And then you go through cycles of these, and at the end of it, what happens is you have those B cells that bind very well, high affinity ones that can go on to become long-lived plasma cells and memory B cells, right? This is sort of like what happens to those of us who are faculty members. Everybody enters, right, with such promise, and we're going to save the world. And then we go there, we mutate, and then as life goes on, we get selected and selected. Very rare of us make it, most of us don't, and that is life. It's the same thing in a germinal center. It's just reflection of life itself. And the germinal center, this I got this from I think one of Jason's sister's slide. The germinal center is a microcosm of Darwinian evolution, and it's evolution steroids. Okay, so in the dark zone, they proliferate, they come back in again, and they get selected, they come back in, they make more of it, they get mutated again, and you have this multiple rounds going, and at the end of it, you have really high affinity B cells that are made, and you have this selection, and it works because you mutate. That's good, but the selection is what makes this very, very efficient. Okay. And now we'll talk about antigen-driven B cell activation, generation of B cell, and serological memory. Okay, so this is like a summary slide that I like to show you. So a naive B cell gets activated, you become short-lived plasma cells, which make antibodies in the short term. That lasts very short time. And this part happens both in T-independent and T-dependent antigen. If it's a T-dependent antigen, then you have this plus this germinal center reaction, where this B cell, purple cell, like the color of my shirt, comes in, and now it undergoes somatic mutation. We make all these different colors. And the red one, that's a high affinity clone, is selected to become enticed to enter the memory B cell pool and into this long-lived plasma cell pool that they become plasmablasts. They go to the bone marrow and they take residence, and they make antibodies for a long, long time. Okay, and so the question is, why do these plasma cells in the bone marrow live for a long time? The reason is because in the bone marrow there are these cells, these stromal cells, that hold this B cell, this plasma cell very carefully. And if this stromal cell lets go of the plasma cell, then that plasma cell dies. Okay? Because they have shown that you can actually take antibodies against BLA4 and VCAM1 and so forth, And if you interrupt this, then the long-lived plasma cells will actually die out. And you also need like other cell types that like eosinophils that make APRIL. These are like growth factors that are required for the plasma cells, and also for an IL-6, which is required for them to survive. So they need to be held and they need to be fed, okay? And if you let them go and they just crumble and they die, they're like some people that you and I know in our families, right? You have to be very careful. They're so touchy-feely that they had to be held carefully all the time. One small thing and the whole world crumbles. It's the same thing with this long-lived plasma cell. And in fact, if you actually make this is APRIL, if you make a BCMA FC, that is, if you mop up all the APRIL that's made, then it dies also. It dies of starvation. Very good. So again, the long-lived plasma cells live because they can bind to they form interaction with the stromal cells, and there are other accessory cells that meet their needs by giving them the growth factors that they require for survival. And in return, what the cell does is it makes antibodies for a lifetime, like 10 to the 4 to 10 to the 5th antibody molecules per second. And so what happens then, so that's one half of it. So you establish your long-lived plasma cells. I also said you make memory B cells. Those memory B cells go and now they become part of your primary repertoire. And when this antigen comes again, oh my goodness, these memory B cells are like pumped up and ready to go. And they make a massive proliferation. They get so much plasma cell and they make so much antibodies that you don't even, and it decimates the pathogen so efficiently that you don't even know that you had actually a clinical manifestation, because your immune response took care of it exceedingly well. Okay, so this is the slide that you have seen in Immunology 101. This is antibody concentration in the serum. This is time. You vaccinate or you first exposure, you get a modest immune response, and this takes about 7 days. The lag is actually pretty long. You get a secondary antigen. The same antigen comes back, and then lo and behold, you get like 2 to 3 orders of magnitude. You make much more antibodies, and you could make it much more faster than you did in the primary immune response. So when you vaccinate, what you do is you are priming the system to make memory B cells and serological memory, so that when the antigen comes again, the antibody that's existing will bind to the antigen, make an immune complex, and then the T cell memory CD4 T cells will have the memory B cells give massive activation and plasma cell generation and 2 to 3 logs greater antibody production in the serum. Okay, and so if you compare primary versus secondary, you have seen this a lot. You have the primary response takes 4 to 7 days. Secondary is 1 to 3 days. The peak response is about 7 to 10. This is 3 to 5. And if you look at affinity, the primary is lower and the secondary is usually higher, because you get them from the pool of your memory B cells. Okay, so this is my last slide. So why are memory B cells better than primary B cells? I used to have like another 10 slides that I go through experiment data, which I decided to take that out. It's because of two things. Number one, the memory B, if you look at, so they did this an experiment where they took a B cell receptor from a memory B cell, and then they made a mouse by, the way they made Dolly the sheep. They transferred the BCR into a new animal, so that every B cell has this memory B cell receptor, highly mutated one. And they wanted to know if they immunized, did they get a secondary response or not? The answer is no, because the memory B cells work because they are epigenetically and transcriptionally distinct from the naive B cell, so that they can actually respond much faster. They can differentiate faster. They have all the things ready to go, and they're wired to respond much faster. Okay, and most importantly, if you need helper T cells, memory helper T cells, in order for a memory B cell response to work. If you experimentally, if you don't transfer in memory B and memory T cells into a new animal and immunize, if you only put memory B, you do not get a massive second response like you do with the when adding the combination. Also, immune complexes are very important. You can actually make an antigen antibody immune complex just by itself and vaccinate and get like a massive immune response as well into a naive animal. And with that, I'm going to stop, and I'm happy to answer questions. And I'm done
Thank you.
I did in 40 minutes.
Yeah, Valentine. Thank you so much. Thank you very much. We have some questions here for you.
Sure.
Um, why are CDRs in antibodies immunotolerant? I mean, why don't CDRs act as epitopes to induce antibody response?
Well, yeah. So first thing is, it's a bit complicated answer, but I'll tell you what I think it is. Okay. So I actually believe the way you make B cell epitopes is you also need a T cell epitope in a near proximity for it to work. Okay. So one, it's possible that these CDRs, they could be immunogenic, but they may not have a T cell epitope nearby in order to provide help for that particular epitope, and that's why it does not work. But having said that, if you look at Niels Jern's old papers and stuff, where they do the anti-EDtypic antibodies and stuff, this should be in fashion like before my time. You can actually immunize and make antibodies against those. So it's not a problem. You can actually do it, but in real life, you hardly make them. Or I don't know if people have actually gone and looked at them that carefully. Probably not.
But for the reason you suggested, Joshi,
Yeah.
it's always hit and miss to make anti-deutypic antibody because you may not have T cell epitope.
Exactly.
Okay. T-independent B cell activation is generally because of polymeric structures. Is it possible for multiple identical monomeric antigens to also elicit a T-independent response by binding several BCRs at a time?
Yeah, actually, so there are two ways you have T-independent. One is, let's say it binds to a receptor on it, like LPS. It's a T-independent. It can bind to TLR4 and activate like a whole bunch of. We will keep that aside. That's not the question. How antigens activate B cells in the absence of T cells is as follows. They have this repeated motif. It'll be like Joshi, 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 Joshi. They'll be like repeated motifs together. And then when a bacteria presents that, it can actually cross-link a multitude of B cell receptors on that B cell and just like get them to get activated. That's the way it has been explained to me.
Yeah. Fundamentally, it's cross-linking which is important.
Exactly. So, and if you look at the repeating motifs, that's what you see in this T-independent antigens.
So why during class switching, the antibodies go from high avidity isotypes to low avidity isotypes? Is it the question is okay?
Yeah. So so when you go from IgM, it's high avidity. You go to IgG, it goes to low avidity. That is true. Okay. So when you increase your affinity so spectacularly, and then also you change your half-life, like IgM lasts for 7 days, the 2A lasts for 21 days. So you can get away with having high affinity, and you don't need to be high avidity in order to bring about the same thing. And evolutionarily, I think all this class switching, everything comes later on, right? So we all start with IgM. At that time, when you don't mutate and you don't make many more of you need to have the high avidity in order to mop up the pathogens. And then when you can make high affinity, then it's a much more surgical strike because you can do it with much more precision.
Okay, a couple more questions. Do plasma cells produce antibodies in absence of antigenic stimuli?
Yes. So it's like the Energizer bunny. That's all it knows how to do. It keeps making it. It just keeps making it over. That's its job. It just makes it. And it does not need antigen. It's like a ball going downhill. That it keeps going and it keeps going, and it does not need antigenic stimulation. Once you become a fully differentiated plasma cell, it will continue to make antibodies. As long as
another question is, do we need to modify the existing vaccines for diseases as the microbes tend to mutate?
You always have to. That's so in flu, right? In influenza, that's the best example. The way it works is, so every February there's this meeting that happens they get together and say, what is, so before we get into flu, if you look at chicken pox, right? The chicken pox that your grandfather and my grandfather saw is very similar to the one that you and I see today. It hasn't changed at all. If you look at influenza, for example, that thing just keeps changing. It mutates, and it's multiple genomes, so it gets shuffled also. So it changes that way as well. And so what happens is, so every year you need a new vaccine. So what we do is in the spring, there's a meeting and they decide and say, of all the different the flu virus that are circulating, which ones do we think are going to dominate in the fall, right? And then we make an educated guess and we make the vaccine. But 60% of the time we are absolutely correct. We match it and then we nail it. 40% time we don't. What we thought would dominate did not dominate, right? So the bacteria also evolve in order to overcome this. So that's when I asked Shiv the question about the COVID vaccine. So right now, only 10% of the people have antibody responses against the virus. So the virus just is not under no pressure to change. But once we start vaccinating and we get more immunity, it's possible that these things could change as well. And then the question is, do we need an annual vaccine just like flu? The mutation rate is nowhere near flu, so we don't have to worry. But nonetheless, it raises the question that's where we may have to go. And remember, these viruses are very clever and they're alive, and they just mutate and they select, and they always come up with a way to get around.
Okay. So how does the Circ protein tyrosine kinase pathway play a role in T cell independent immune response?
That signaling is one of my weak points. I think Shiv would know about that much more than I, because he spent a lot of his lifetime studying those very carefully. So if he's still on, perhaps
Yeah,
he
I'm
could take
still
a
on. I have to leave in about 5-10 minutes, but I'll let you answer all your questions and I can come back to that.
All right, cool.
Okay. So
All right, next one.
The next one is about pollen allergens.
Yes.
Pollen allergen can induce secondary response for generating sustained IgG antibodies from plasma cells migrated in bone marrow. Is it so?
I don't know. I have to look that up. I haven't thought about that. I always thought they were IgE, and then you would make IgG if you took the desensitization shots, so that you'd switch from the IgE to an IgG response, so that you would mop up the pollen, so that you do not. So your mast cells are covered with IgE. Let's say it's specific for pollen. And if you vaccinate this person with the pollen antigens, you make IgG. The IgG will actually go mop up all the pollen antigens, and then your mast cells don't have enough antigens in order to cross-link them and degranulate.
Okay.
That's what I can think of the top of my. I could be wrong about this.
Okay. So I think that's about it from. And Shiv, would you like to come back to that?
I'll respond to both those things. Yes, Joshi is right. In humans, there's good data for IgG4 being induced instead of IgE, and that will compete. And for the signaling question, always when you activate the BCR, you go through first Sark family kinase about 1 minute later, which will phosphorylate the ITAMs of Ig alpha and Ig beta. Then you will activate SIC roughly like 2 minutes after you activate. SIC is like ZAP70, it has 2 SH2 domains. Then you'll get the activation downstream of PI3 kinase, you'll activate BTK. There are 3 different tyrosine kinases needed. Even with T-independent responses, you need all 3 to get your response. So in the experiments having done in the mouse, if you lose BTK, your T-independent response is screwed up a bit.
Okay.
And I have a question for Joshi, which I put in the chat before. Joshi, did you cover affinity? I'll have to leave soon, but did you cover affinity being much lower in memory compared to plasma cells?
No, I didn't.
Okay, so I mean, that's a phenomenon, right? That you come out with the much broader repertoire, which is lower affinity, but plasma cells. Okay. All right, great talk. I'm sorry I have to leave.
Oh, I think we are about done. So thank you very much, both the speakers. So much. Thank you so much.